For those of you who do not know who I am, my name is Mark Mofis. And like Fernando said, my presentation is when cholesterol in the body goes from necessary to life with me. And I'd like to thank my mentor, Dr. Rollins, for all the help. So let's get started. So to give a brief outline of what I'm going to talk about in this presentation, first I want to talk about what exactly cholesterol is. Secondly, I want to talk about the different ways that cells can obtain cholesterol. Next, I want to talk about a good level of cholesterol to have in the body. I want to discuss a phenomenon known as atherosclerosis. And then lastly, I want to propose an ideal level of cholesterol in the body. So, what is cholesterol? We know that cholesterol is a very waxy, lipidy substance, and most people think cholesterol is bad, but cholesterol is actually good for you. And cholesterol is just an organic molecule that the body produces. As you see here, cholesterol has 4 big, bulky rings, which make it a steroid, and cholesterol has a hydroxy group, which make it an alcohol. Because it's a steroid and an alcohol, it's a sterol. And cholesterol is actually needed by the body. So, what does cholesterol do in the body? Cholesterol has a variety of functionings in the body. For starters, we're going to start with the cell membrane. Cholesterol is a very important part of the cell membrane because it helps it maintain its fluidity and it makes sure that certain stuff doesn't enter it. Cholesterol is also a very important precursor for steroid hormones. It helps produce hormones like testosterone, aldosterone, cortisol, and cholesterol even helps convert sunlight into vitamin D. Cholesterol is also very important in myelin sheaths. As you see, some nerves can be unmyelinated and myelinated. Myelinated nerves are like right here. They have a myelin sheath surrounding it. Cholesterol is a very important part of that sheath, and that actually helps the nerve transmit its action potential at a faster rate, and it also protects the nerve. Lastly, cholesterol is a very important component of bile acids, and bile acids are what allow digestion of lipids. So, cholesterol is very important in the body. So, there are two ways a cell can obtain cholesterol. The first way is through endogenous synthesis, and the second way is through endocytosis of LDL. So, in endogenous synthesis, acetyl-CoA joins with acetyl-acyl-CoA, and that forms HMG-CoA. HMG-CoA is then reduced by HMG-CoA reductase, and that forms mevalonate, through a series of steps that eventually yields cholesterol. The second way is endocytosis of LDL. And this basically means that the cell obtains its cholesterol from an exogenous source. But before we can talk about this, we have to first talk about what exactly LDL is. So, to talk about LDL cholesterol, we have to talk about the nature of cholesterol. Cholesterol as a molecule has many carbon to carbon bonds and carbon to hydrogen bonds, which means it's hydrophobic, it's not soluble in water. As you guys may know, the blood is 55% plasma, and plasma is 92% water. So if cholesterol is not going to be soluble in water, it's not going to be soluble in blood. So the body needs a way to overcome this. The body does this by having different types of lipoproteins. Lipoproteins essentially transport the cholesterol someplace without getting stuck in the blood. LDL is just a type of cholesterol. So there are a few types of ways that cholesterol can be transported, but the two main ways are through HDL cholesterol, which is high density lipoprotein, and low density lipoprotein, which is LDL. And we're mostly going to focus on LDL. Like said previously, cholesterol is very hydrophobic. And in this inner region, this is where cholesterol is. Even though cholesterol is very hydrophobic and it's not soluble in water, cholesterol is completely covered by a hydrophilic region, which is a phospholipid layer. These phospholipid heads are hydrophilic, which gives cholesterol the ability to be transported in water. Apolipoprotein B is a protein on the surface of LDL, which allows cholesterol to be endocytized, like we talked about earlier. This protein is very important, as you'll see in the following slide. So here is LDL cholesterol, and here is apolipoprotein B. And here is the cell. That's the nucleus, and that's the receptor on the cell. The receptor on the cell is what allows the cell to endocytose cholesterol. So the receptor, as you see, is like this. It joins to the apolipoprotein B protein, and it then takes the entire complex into the cell. The entire complex is then internalized into the cell, including the receptor. lysosomes are in cells, and as you guys know, lysosomes are important for degrading stuff. So the lysosome actually interacts with the complex. And inside the lysosome completely degrades the LDL through its digestive enzymes, which yields free cholesterol in the cell. And the receptors are then brought back to the surface. So now that we discussed that cholesterol has many functions in the body, cholesterol is very important. But how much cholesterol is too important? And to answer this question, like how much cholesterol is too much in the body, we need to talk about a phenomenon known as atherosclerosis. Atherosclerosis is a buildup of plaque and fatty material on arterial walls. And this may have, people don't really know the causes of this, but the most predominant cause is high LDL cholesterol, as you will see. So no one really knew what caused atherosclerosis until 1914, where a scientist named Aniskov actually did experiments on rabbits where he fed rabbits a high cholesterol diet. And he saw that after he fed these rabbits a high cholesterol diet, these rabbits actually had fatty streaks. They developed atherosclerosis. So here are some of the results from a similar experiment. Basically, in this experiment, what they did was that they fed rabbits a high cholesterol diet. There were the regular rabbits, and then there were rabbits that they fed a high cholesterol diet. And they saw that when they fed the rabbits a high cholesterol diet, those rabbits had a very high plasma cholesterol level, and those rabbits also developed atherosclerosis. As you can see in a different experiment, they fed rabbits again a high cholesterol diet, and they saw that in the intima of the arterial wall, right here, there were a lot of lipid deposits in the intima, which meant that the cholesterol was entering the walls. And in pictures B and C, they even saw foam cells, which are basically just macrophages engulfed with lipids, and these were all in the wall. So a lot of people did do experiments to see that high cholesterol gave atherosclerosis, but next they wanted to see exactly how much cholesterol it took to induce atherosclerosis. That was seen in this experiment. They saw that when they fed rabbits a dietary cholesterol amount between 0.05% and 0.15%, their cholesterol levels in their blood stayed relatively stable, and the total cholesterol content is directly related to atherosclerotic progression. So when feeding the rabbits dietary cholesterol this low, they saw that they did not develop atherosclerosis. It wasn't until the dietary cholesterol level that they fed them was 0.20% or higher that their cholesterol levels began to skyrocket. And in these same rabbits, they saw development of atherosclerosis. So here's the same study, but here they checked it out by using the Sudanophilus stain surface. Basically, Sudan4 is a stain, and it stains for lipids. So if there's a high concentration of this stain, that meant that there's a lot of lipid staining in the arterial walls. In the same experiment, from 0.05% and 0.15%, there isn't much staining of lipids, which meant at this diet, there wasn't much cholesterol in arterial walls. But at 0.20% and higher, there was a high amount of lipid staining, which meant a lot of cholesterol entered the cell walls. So now that we saw that atherosclerosis is in the body and how it works, let's talk about exactly the mechanism that induces it. So here is the blood. There are red blood cells in the blood, there's LDL cholesterol, here's the endothelium, here's the tunica intima, and here's the tunica media, which smooth muscle cells are found. LDLs can enter the tunica intima because these endothelium are dysfunctional. In the tunica media, the endothelium actually release reactive oxygen species. These reactive oxygen species actually oxidize the LDL. It's important to note that when the LDL oxidize, they cannot leave the tunica intima. After the LDL oxidize, they actually will trigger the endothelial cells to release adhesion molecules on their surface. These adhesion molecules allow floating monocytes to enter the tunica intima and differentiate into macrophages. When they differentiate into macrophages, these macrophages actually scout and look for oxidized LDL, and then they engulf them. And when they engulf them, the macrophages become foam cells, which are basically macrophages full of lipids. These foam cells do a variety of things. One of the things they do is that they release chemokines, which actually attract more macrophages to the area. Secondly, they release a growth factor called IGF-1. IGF-1 causes the smooth muscle cells in the tunica media to proliferate and migrate into the tunica intima. These smooth muscle cells in the tunica intima release a lot of collagen, which is what is responsible for making the atheroma a lot harder. Foam cells also, they can die, they will die. So when the foam cells die, they release a lot of lipid droplets in the cell, or in the artery wall. And all of this happening together contributes a lot of inflammation. So as this is happening, LDL is still entering the intima, macrophages are still coming, so this makes this area grow bigger and bigger and bigger, and this leads to a plaque. And I know it's really hard to see in this diagram, but I can show you guys on a vessel or an artery. Here's how an artery should look like. The red blood cells should flow easily. There should be no inhibition or obstruction of flow. But when atherosclerosis occurs in the artery, it begins to grow into the blood vessel, and blood there's minor inhibition for the blood. If it was to ever rupture, there would be a thrombus. This will begin to clot, and normally endothelial cells release anti-clotting factors, but because these endothelial cells are dysfunctional, they don't do that, so this clots. The blood gets stuck here, and any tissues or cells beyond this point don't receive oxygen because there are no red blood cells. So this is very dangerous, especially if it happens in the heart. If the heart muscle does not receive oxygen, that will lead to myocardial infarction, otherwise known as a heart attack. So let's talk again what's the cause of atherosclerosis. We know that atherosclerosis is caused by high LDL, high cholesterol, because we saw that in the studies. Here's another study which showed that, same study, when they fed rabbits a high cholesterol diet, those rabbits developed atherosclerosis in the arteries. Atherosclerosis can be seen right here, here, and here. And it's not just rabbits that they fed the high cholesterol diet. They did many different animals, like rabbits, mice, pigs, and even non-human primates. Non-human primates are very similar to us, and they saw that in these results, they showed that when they fed these non-human primates high cholesterol diet too, they developed atherosclerosis. So, like I said previously, we need cholesterol, but how much cholesterol do we need in our diet? These are the current National Cholesterol Education Program guidelines, and they believe that anything less than 200 milligrams per deciliter is desirable for cholesterol, and anything higher than that would be too high. In this slide, you don't have to be all of this, but the purpose of this slide is that people typically have higher levels of cholesterol than the guidelines. But the biggest point I'm trying to make is that even though people do have high cholesterol levels, the 20% of people, like cardiac events that happen between these high cholesterol levels are in the abnormal ranges. That leaves 80% of high cholesterol events happening to people who have normal levels of cholesterol. So I believe that normal might not be the best level of cholesterol. We need to update that. So here's a different study where they compared wild animals with wild mammals, wild primates, and even different like human populations. And they saw that in all these people, all these animals, they had half the level of cholesterol that we did, about half. It was only in adult Americans or people from first world countries that the cholesterol level was on average 208 milligrams per deciliter. It should also be noted that in all these besides the adult American, they do not develop atherosclerosis. Atherosclerosis is not seen in these people. Here is another study. They decided to see the correlation with LDL cholesterol and the mean luminal diameter. And they saw that depending on someone's cholesterol, they would have, if someone had a high cholesterol, like there was more atherosclerotic progression, but the lower the LDL cholesterol, the lower the progression. And if you follow this progression line, you will see that if someone's LDL cholesterol was less than about 67 milligrams per deciliter, no atherosclerosis does not progress. It does not happen in the body. So, in conclusion, the point I'm trying to make is that we definitely need cholesterol. We need cholesterol for our bodily functions. Like, cholesterol is necessary for us to survive. But the cholesterol of the typical cholesterol guidelines, the typical normal amount of cholesterol, it leads to atherosclerosis. Atherosclerosis leads to heart disease, and heart disease is the biggest killer worldwide. It kills 600,000 people a year in America. 1 in every 4 deaths from heart disease. So even though we do need cholesterol, the cholesterol that we currently aim to get is a little too high. So there needs to be an ideal physiological range where at the low end of the spectrum, we don't have enough cholesterol for our cells to operate. And at the high end, we have too much and we develop atherosclerosis. According to the studies and the data I've seen, I believe that the ideal range is to have an LDL cholesterol about 70 milligrams per deciliter or a little bit lower. Okay, that's that.